Welcome back as we continue our journey through this process of cellular respiration. In this part of the chapter, we're actually going to see how the splitting apart of glucose molecules that we saw last time is ultimately going to lead to the synthesis of ATP to power all of the work that is done by cells. So when we last saw our glucose molecule obtained from food, it had been split through the process of glycolysis into two molecules of pyruvate. So we've gone from a 6-carbon sugar down to two 3-carbon smaller chains of pyruvate. And in the process, we generated two molecules of NADH. So electrons were transferred from the glucose derivative to reduce NAD plus to NADH. And then the other result of that was the substrate level phosphorylation of a couple of molecules of ATP. So this is where we're going to pick up on the process. We've got pyruvate in the cytosol, and now we need to move it on into the next step. So the next thing that needs to happen before these 3-carbon molecules can enter the next phase, which is the citric acid cycle, they need to be modified to be able to be used by the citric acid cycle. So pyruvate is going to be oxidized, and then the harvesting of the energy in these carbon molecules is going to be completed. So in the presence of oxygen, pyruvate is going to enter the mitochondrion in the case of eukaryotic cells, where the oxidation of glucose is going to be completed. So we're going to finish harvesting all of the energy from those molecules that came from food as we go through the rest of this process within the mitochondria. So we're going to go now into the mitochondrial matrix. So before the citric acid cycle can begin, pyruvate needs to be converted into a form that can be used by the citric acid cycle. And so pyruvate is going to be converted into acetyl coenzyme A, which is going to be the link between glycolysis and the citric acid cycle. This step is carried out by a multi-enzyme complex that catalyzes these 3 reactions. So first, pyruvate needs to enter the mitochondrion through a transport protein. So the first reaction is the oxidation of pyruvate and the release of CO2. So at this point in the process, we want to start really keeping track of where these carbon molecules go. We want to track the carbon as well as tracking the electrons. So we're going to release a carbon dioxide. So now instead of a 3-carbon chain, we're going to have carbon dioxide in a 2-carbon chain. This is going to be accompanied by reduction of NAD plus to NADH. So here are a couple of electrons that we want to keep track of. We've got the production of some more NADH. And then finally, we're going to take the remaining 2-carbon fragment that came after we took off the CO2. Remember, we started with a 3-carbon chain, we removed a CO2, and this is what we have left. And that is going to be combined with a substance called coenzyme A to form acetyl-CoA. So this is an acetyl group, the 2-carbon chain, attached to coenzyme A. We call it acetyl-CoA. So during these reactions, our inputs are pyruvate, NAD+, and coenzyme A. In the end, what we end up with is carbon dioxide. So this is the first of the carbon dioxide molecules that are removed out of the system as waste, NADH, and then acetyl coenzyme A come out, acetyl-CoA. So this whole pyruvate processing is also regulated by feedback inhibition, just as we saw with glycolysis. So when the products of glycolysis and pyruvate processing are abundant, one of the key enzymes that run the pyruvate processing is going to be phosphorylated. It's going to change its shape and be inhibited. So now our carbons, now in the form of acetyl-CoA, are going to go and enter the citric acid cycle. So each acetyl-CoA from pyruvate processing is going to be oxidized in this process of the citric acid cycle into 2 carbon dioxide molecules. So that's where these remaining 2 carbons are going to end up through this process. The citric acid cycle is going to be located within the mitochondrial matrix, so inside that inner membrane of the mitochondrion in eukaryotes, and in the case of prokaryotes, it's going to happen in the cytosol. And these reactions are organized into a cycle. So let's see how that works. The acetyl group of acetyl-CoA is going to join the cycle by combining with oxaloacetate to form citrate. So then the next 7 steps of the citric acid cycle are going to result in the conversion of this citrate back into oxaloacetate, making the process a cycle. So the output from one turn of the cycle is going to be one of the 2 inputs for the next turn of the cycle. And the cycle as a whole is going to oxidize organic fuel derived from pyruvate, generating 1 ATP by substrate level phosphorylation, as well as a bunch more of these electron carriers, so 3 NADH and 1 FADH2 per turn of the citric acid cycle. And these are going to be the molecules that are going to relay the electrons extracted from the food to the electron transport chain. So all of this is about harvesting those high energy electrons, converting them into a little bit lower energy electrons with each transfer to another receiver. Again, this is a lot of information on this chart. I don't want you to worry about the names of the intermediates or the names of the enzymes that do this or the structures, but I do want you to notice what's happening to each of these carbons as it goes through the cycle, and at what point you get these products of carbon dioxide, NADH, and FADH2. The acetyl coenzyme A, which remember is a 2-carbon chain attached to a coenzyme, is going to come into the cycle and join up with the oxaloacetate, which is coming back from the last turn of the cycle. And this is color-coded so that you can see where these carbon atoms are coming from and where they're going. So the ones that are coming from this new acetyl coenzyme A that's joining the cycle are coded in blue. The ones that are eventually going to be removed as carbon dioxide are going to come from the last turn of the cycle, and they're color-coded in red. So now we've got this 2-carbon chain joining up with this 4-carbon chain to go back to a 6-carbon chain in the form of citrate. So in this 3rd step of the process, we're going to remove another carbon dioxide from the 6-carbon chain, bringing it down to a 5-carbon chain. And in the process of doing that, we're going to generate another NADH. So oxidation, reduction, coupled together in this one step. We're removing the carbon dioxide as a waste product. We're storing another pair of electrons in this NADH. Again, in step 4, we're removing another carbon dioxide as waste. We're going again from a 5-carbon chain back down to a 4-carbon chain, oxidizing carbon to CO2, reducing NAD plus to NADH for our second NADH of the cycle. In the 5th step, we're going to do substrate level phosphorylation, either producing ATP or GTP, depending on the situation. GTP is a similar molecule to ATP and can perform many of the same functions, although not storing quite as much energy as ATP does. In the 6th step of the cycle, we're going to have another reduction, this time of FAD to FADH2. As we continue to convert this carbon chain, we come around to the final step, step 8, we get one more reduction reaction converting NAD plus to NADH, storing another one of these electron pairs to be transported to the electron transport chain. And so if we continue to track these blue carbons that came in from this cycle, one of the 2 of them is going to end up being destined to be removed in the next round. The other one is going to continue to be part of this chain for one more cycle. So now let's think about how this process is regulated, and this one is also regulated by feedback inhibition, but it has a number of different sort of breakpoints that can be influenced to slow down or stop the citric acid cycle when ATP is abundant. There are a couple of steps that are regulated by feedback inhibition by NADH and also ATP. So if we get an excess of either of those molecules, they're going to regulate the enzymes at steps 3 and 4 to slow down or stop this process. And then also the first step in the cycle is regulated by ATP. So if ATP is abundant, it can stop it just at the point of acetyl-CoA joining the citric acid cycle to shut the whole thing down. So reaction rates are high when ATP and NADH are scarce. Reaction rates are going to be low when ATP or NADH are abundant. So when the products of the whole set of reactions are abundant, we're going to slow it down and create less product. When the products are scarce, we're going to keep it going to produce more of those products. So now let's see what we've gotten out of this whole process. So we've gotten a whole bunch of NADH and FADH2. So for each molecule of glucose that's oxidized so far, we've got the 6CO2. One of those comes off for each of those pyruvate molecules. Remember, we got 2 pyruvate molecules for each glucose. So one comes off of each pyruvate to get 2 carbon dioxides at the point of pyruvate processing. And then we get 2 more for each turn of the cycle, but remember each glucose gave us 2 pyruvates, so 2 turns of the cycle for each glucose, giving us a total of 4 carbon dioxide. And then for NADH, we get 2 during glycolysis, 2 more during pyruvate processing, 1 for each of those pyruvate molecules that comes from each glucose. And then we get a total of 6 NADH and 2 FADH2 for the 2 turns of the cycle that it takes to process a single glucose molecule. So a total 6 carbon dioxide, which is going to be disposed of when you exhale, it just diffuses right out of the cell. 4 ATP from substrate level phosphorylation during both glycolysis and during the citric acid cycle, and we're going to get a total of 10 NADH and 2 FADH2 from this process. So now what's going to happen to that NADH and FADH2? We need to take those electrons and do something with them. So what these are reduced electron carriers. So we need to make sure that we pass those electrons on, and we also need to make sure that we're regenerating the oxidized forms, the NAD plus and FAD, so that they can continue to do this work of transferring electrons. So let's see how that happens. So most of the glucose's original energy is contained in those electrons that are transferred to NADH and FADH2 at this point. In that transfer into these carrier molecules, they have not lost very much of their energy. They still have most of the energy that was originally contained in those electrons. So the electrons, along with the protons that came with them, are ultimately going to be transferred to oxygen to form water at the end of this whole process. So 2 questions remain. What happens to all that energy that's released as the electrons are transferred from carrier to carrier through this process? And then how is that energy used to make ATP? We can't use it directly. Energy tends to be just dissipated if it's not transferred to something else. So how do we end up with ATP out of this whole process? So now we're going to take a closer look at that part of this problem. During oxidative phosphorylation, a process called chemiosmosis is going to couple electron transport to ATP synthesis. So the transferring of electrons is not going to synthesize ATP directly. These 2 processes have to be coupled in some way to be able to harness that energy so that it's not just dissipated as heat. At the end of the citric acid cycle, so following glycolysis and the citric acid cycle, NADH and FADH2 account for most of that energy that's been extracted from food. It's just been transferred to these 2 carrier molecules. Back when this was first being investigated, researchers determined that NADH is oxidized when combined with the inner membrane of mitochondria in eukaryotic organisms. In prokaryotes, it's oxidized if it is put in solution with a plasma membrane. So we know that it's something about these membranes themselves, things embedded in the membrane, that's going to cause this oxidation of these carrier molecules back to their oxidized form, NAD plus and FAD. So molecules in the inner mitochondrial membrane can cycle between oxidized and reduced states. So they're basically going to accept that electron and then pass it on to something else. So reduced when it accepts the electron, oxidized again when it passes it on to the next molecule in the chain. So what are these molecules and how do they work? So let's take a look at the electron transport chain. So the molecules that oxidize NADH and FADH2 are called the electron transport chain. You can think of it as a bucket brigade. Most of these are proteins that are easily oxidized. So one of the elements in this electron transport chain is a lipid-soluble non-protein called ubiquinone, or coenzyme Q, sometimes just called Q. So it's lipid-soluble, which means it can actually move through the hydrophobic tails of that phospholipid bilayer, and it acts to shuttle these electrons between protein complexes that are embedded in the membrane. So the lipid solubility is really important. This is where we see the importance of the fluidity of that phospholipid bilayer so that these molecules can actually move back and forth through the middle of that phospholipid bilayer. And they have different abilities to accept electrons called their redox potential. Some of them are going to accept only electrons. Others are going to accept electrons along with their accompanying protons. So this is the really important point. Electrons are going to drop in their free energy as they go from one carrier to the next down the chain. So what does this mean? This means that each element in this electron transport chain is going to be a little bit more electronegative than the element before it. So that means that that next element in the chain is going to hold those electrons a little bit more closely to the nucleus. It's going to be a little bit lower energy, and then the reason it's going to give up those electrons is because the next molecule in the chain is going to be even more electronegative, and those electrons are going to be even more stable and have lower energy when they get to the next member of the chain. So this is going to happen all the way down, dropping small amounts in free energy as it goes to more and more electronegative carriers until it ultimately gets down to the most electronegative of them all, oxygen. It's going to combine with free protons in the mitochondrial matrix to form H2O. And so these electron carriers are going to alternate between reduced and oxidized states as they accept and donate electrons. Every time they accept an electron from the molecule before it in the electron transport chain, it's going to be reduced. Then when it donates that electron to the more electronegative element in the chain downstream of it, it becomes oxidized. So it's constantly going back and forth between an oxidized and a reduced state. So here's an image of what this looks like. So initially, these electrons are going to be transferred from NADH, and this is going to be at the very beginning of this electron transport chain where NADH gives up its electrons. It's going to be transformed back into NAD+, and it's going to go to the proteins in complex 1. FADH2 is going to donate its electrons into complex 2, which is a little bit further down the chain. It's going to miss a couple of these energy extracting steps, so actually less ATP is going to be generated per molecule of FADH2 than will be generated per molecule of NADH. So these electrons are going to be transferred. Q is going to be, remember, that lipid-soluble molecule that's going to actually move through the cell membrane, through those hydrophobic tails, and take electrons both from complex 1 and complex 2 and shuttle them over to complex 3. So we've got a series of electron carriers, cytochromes, and some iron and sulfur-based electron carriers down through complex 3. There's going to be another little shuttle protein, cytochrome C, that's going to move it between complex 3 and complex 4, losing energy. So remember, we're on a graph now as we tumble down here of free energy, going from higher states of free energy down small little steps to lower states of free energy until finally it takes that final tumble of energy down to oxygen, which is going to combine with hydrogen ions in the matrix to form water. So electrons, again, pass through a number of proteins, including cytochromes, which include an iron atom, sort of similar to hemoglobin, and eventually landing on oxygen. And the thing to notice is that ATP does not come into play anywhere in this figure. There is no ATP directly generated by this release of energy. So what does it do? It breaks the large free energy drop from food to oxygen into smaller steps that releases small amounts of energy at a time. But what happens to that energy? How does that energy end up being made into ATP? So this is where this energy coupling mechanism comes into play, chemiosmosis. So the energy that's released as these electrons are passed down the electron transport chain is going to be used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Remember when we talked about the endosymbiont hypothesis, about that double membrane in mitochondria, why it's important to have those separate chambers? This is why. We need this separated chamber to hold this high concentration of hydrogen ions separated from this electron transport chain to create an electrochemical gradient of protons. So we're pumping hydrogen ions against their concentration gradient using the energy that's being released from this electron transport chain. So most of that chemical energy at this point that came from the glucose, that came from the food that was eaten, is now accounted for by this proton electrochemical gradient, which contains a large amount of potential energy. It's both a concentration gradient, but remember also a voltage gradient because each of those protons has a positive charge. So we call this electrochemical gradient the proton motive force. What is the proton motive force going to do? So the only way for these hydrogen ions to get out of the intermembrane space is through intermembrane proteins that synthesize ATP. So the protein complex is called ATP synthase, and that is the only way for hydrogen to get through. This is an image showing you this electron transport chain generating energy that is pumping these protons, these hydrogen ions, out into the intermembrane space, creating a high concentration of protons in the intermembrane space, but also a relatively positive voltage, relatively negative voltage inside the matrix, and low concentration of hydrogen ions. Let's take a closer look at this ATP synthase molecule. It's very cool and very complex. So the ATP synthase consists of two components. There's an ATPase knob. This is where the actual reaction is going to happen down here in the F1 unit. Then there's a membrane-bound proton transporting base. So we call this the F0 unit or F0 unit. So F1 unit down here, F0 unit. And the units are connected by a shaft. So there's a rotating shaft and also an element called a stator that's going to connect the two and hold them in place. So this F0 unit is a rotor. It's actually going to physically turn as the protons flow through it. So one proton at a time is going to attach to binding sites on each of these elements of the rotor and make a complete turn around the rotor. So the spinning changes the conformation of the F1 subunit so that it's going to phosphorylate ATP. So this is going to be a mechanical force that's going to power the synthesis of ATP. The energy to produce ATP actually comes from that proton gradient. The energy to create that proton gradient came from the energy that was released as the electrons moved down the electron transport chain. And this process is what's called oxidative phosphorylation. So this is different from the substrate level phosphorylation that occurred during glycolysis. Remember, in the substrate level phosphorylation, a phosphate group that was attached to another molecule was transferred to ADP to create ATP. In this case, the free ADP and inorganic phosphate that's present in the mitochondrial matrix is just put back together again in oxidative phosphorylation. So it's a matter of where the phosphate comes from. If it comes attached to another molecule, that's substrate level phosphorylation. If it's inorganic phosphate that's free in the mitochondrial matrix, then that's oxidative phosphorylation. So most of the ATP made during cellular respiration is made by this process of oxidative phosphorylation. That's where we get most of the energy out of those food molecules, it's contained in those reduced NADH and FADH2 molecules that then pass that energy down the electron transport chain. So let's take a look at our net gain here in cellular respiration. So during cellular respiration, most energy flows in this sequence. So from glucose to NADH to the electron transport chain, which is going to generate this proton motive force to create ATP. About 34% of the energy contained in a glucose molecule is transferred in this way to ATP through all of these intermediates during cellular respiration. And this is going to net about 32 ATP per glucose molecule. That's going to vary somewhat for different reasons, which we will get to in a moment. So 34% efficiency is not great. This is why we generate heat, and the rest of that energy is lost as heat. So there are a few reasons why we can't know exactly the number of ATP that we're going to get from a glucose molecule because it's not going to be consistent. So one of those reasons is that the process of phosphorylation and these redox reactions, the transfer of the electrons, is not directly coupled. It's not the direct result of the redox reactions. They just provide the energy that's going to fuel the oxidative phosphorylation. And so the ratio of NADH to ATP molecules is not a whole number, and it's not necessarily even going to be consistent. The second reason is that the ATP yield is going to vary depending on whether electrons are passed to NAD+ or FAD in the mitochondrial matrix. NADH and FADH2 cannot actually move from the cytosol into the mitochondrial matrix. They actually pass their electrons to electron carriers in that cell membrane to transport them across the membrane, where they are picked up by another NAD+ or FAD molecule. And which one of those picks them up is going to determine where in the electron transport chain they deposit those electrons, which is in turn going to determine how much energy is going to be extracted from those electrons. So finally, number 3, this proton motive force is not just going to be used for synthesizing ATP. It's going to be used to do other work as well. And one example is moving pyruvate molecules into the mitochondrial matrix. That's going to happen using the proton motive force for active transport across that membrane. So depending on what other things that energy is being used for, more or less ATP can be produced using that proton motive force. So this 32 ATP is an estimate. It's actually perhaps a maximum number of ATP that you can get per molecule of glucose. So another thing to keep in mind is this estimate of 32 ATP depends entirely on the electrons going through this electron transport chain in the presence of oxygen, oxygen being that final electron acceptor that's going to pull the electrons down through this electron transport chain. If oxygen isn't present, then we get a completely different accounting for the amount of ATP that we can get out of glucose, depending on what kind of process is going to be used for getting that energy. So that's going to be the topic of the next web lecture: what happens in the absence of oxygen to all of these processes.